Welcome to another episode of the Dendrite podcast with Farnum and Nika. This is a conversation with Dr. Vahab Soleimani. He's a professor at the Department of Biochemistry, Microbiology, and Immunology at the Faculty of Medicine at uOttawa. He's also an affiliate investigator at Ottawa Hospital Research Institute.
During his scientific career as a researcher at University of Ottawa and McGill University, he has focused on the effects of mobile DNA elements on the evolution of genome and molecular basis of myogenic differentiation and muscle stem cell function during his master's and postdoctoral fellowship. His current research focuses on regenerative medicine and muscle stem cell biology. We hope you enjoyed this episode, and now here's Dr. Vahab Soleimani.
Hello, Dr. Soleimani. How's it going? How are you today?
Good.
Thank you for coming to our podcast. We're really honored to have you here.
My pleasure.
Thank you very much. Let me ask you a little bit about your background. How has it led you to come here and study at University of Ottawa and do things that you're doing right now?
So I graduated from University of Ottawa. I actually came to Ottawa in 1989. Emigrated here, so that's 36 years ago. And then so I settled here and I went to, completed my undergrad degree here. Went on to do a PhD in the same university. And I guess I was too comfortable in Ottawa. I also did a postdoctoral fellowship here at the Ottawa Hospital Research Institute. That was by 2013. At that time, I was looking for PI positions, research positions in various universities. So I ended up in Montreal at McGill University. And I was there for 9.5 years, and then I came back to Ottawa again. So here I am, first as a student and now as a faculty at the University of Ottawa.
Would you ever want to go somewhere else or teach somewhere else other than Ottawa?
I mean, that's a tough question. I spent almost all my adult life in Ottawa, so it's almost like a hometown for me. Okay. Yeah, except for the winters,
Yeah, Ottawa, especially the hockey team's going crazy. I'm so excited. We won last night and hopefully we're going to win for the 5th game. So that's cool. How did you find it? How did you find out that you're interested in muscle research and muscle regeneration? Was it like a sudden thing or was it like a more...
No. My undergrad was I was studying basically molecular biology and genetics with specialization in the program that the university at that time was called biotechnology specialization. And so there, this is many years ago, recombinant DNA technology was pretty cool. Being able to move pieces of DNA around and then trying to figure out gene functions through that kind of technology. It's like an old technology now, but it used to be new and exciting. So this is where my interest came in studying how genes function, how genomes evolve. My PhD thesis was actually on the role of transposable elements on the evolution of genomes using plants as a model. Because they have large genomes, they have a lot of repetitive DNA, which used to be called junk DNA. Of course, now we know that it's got a lot of functions there. So my interest in stem cell biology, certainly, when I was looking for postdoctoral positions, I was open to change the field or the model of studies, either slightly or completely. This is not unusual for people to do that. And one of the labs that I was very interested in working with was right here in Ottawa. It's Dr. Rudnicki's lab, who happened to work on skeletal muscle stem cells. So ever since then I became very fascinated with this model and this organ and how it functions. And so here we go now.
What was the thinking process when you were an undergrad? Were you knew that you were kind of interested in that field, or?
I was always interested in research, not because my parents were scientists or anything like that, but as a child I was very curious. I loved going to nature and then just trying to look around, see how many things we have, organisms, plants, and stuff like that. And also I was very interested in just basically the world around me, and as a kid, kids are by definition scientists, right? So their lab is their mouth. They pick up things, they put it in their mouth, try to figure out how it tastes and what it is. And then some of us, we go a little bit further and then we make that into a career. And certainly this is what happened to me. So I became a scientist, just a continuation of my childhood curiosity.
So could you tell us a bit more about like sarcopenia and cachexia, because these two diseases are, I believe,
Yeah. So these are two. I mean, they're muscle wasting conditions, both of them, but they're caused, but with different reasons. Cachexia is a muscle wasting disease that is the result of many things. Like it could be cancer, could be heart failure, could be COPD. Chronic obstructive pulmonary disorder. I hope I said it right, and many other conditions. So in our lab, I mean, cachexia is an area that we started working on as a kind of a side project a few years ago, and this is now one of the projects that is in the lab. And we're specifically interested in cancer cachexia. So a lot of cancer patients, not all of them, but a lot of cancer patients, especially in the terminal stages of their disease, they have this massive loss of muscle mass and function. And then so what happens is that because muscle is such a critical organ in the human body, not just with respect to movement of your body, but it's because it's oxidative tissue, so it's involved in energy metabolism. It's also the largest endocrine organ in the human body. Not too many people know that. It actually produces a lot of growth factors. So when you have that kind of a dramatic reduction in your muscle mass and function as a result of cancer cachexia, and of course we know a little bit about how this happens, but it's like a tip of the iceberg in terms of the complex nature of how cancer interacts with the body, and then why you lose muscle on such a dramatic scale. So that's the cachexia. Sarcopenia is also a condition where muscle is lost, but it's age-related, right? So we when we were young, and by young, I mean up to 30, muscle is pretty healthy. Muscle growth is normal. Actually, starting at 30 and plus, you see that the signs of slowing down in muscle. But sarcopenia is accelerated in older age, when you're 60, 65 plus. Then you have this accelerated rate of muscle loss.
So for aging, it kind of makes sense for me, but for cancer, usually when we have cancer, we have like upregulation of cell division. Why does it lead to having less muscle?
Yeah, that's a very good question. And the short answer is we don't completely know. The longer answer would be that cancer, by definition, is preoccupied with its own growth and well-being, doesn't care about the body. It uses your body as a source of food. So our thinking is that cancer is actually sending some signals to the body. We have some evidence, but this is very preliminary. I don't mind talking about it without getting into too much details. So we think cancer is actually sending some starvation signals to the body to prevent the body from using nutrients. And perhaps this is the reason why nutritional supplements actually don't help patients that much. And then so because the cancer wants to take everything, right, all the nutrients. So that's probably one of the things that the reasons why you, even in the case of taking a lot of food and nutrients, your body is not able to recover that lost muscle.
Right. Interesting.
So how does muscle stem cell actually contribute to these diseases?
So muscle stem cells, these are a rare population of cells that were discovered in 1960s and early. I think the first publication was by Alex Muro in 1961. That's a long time ago, right? And then from then on, there has been extensive amount of research on these cells because of the central role that they play in muscle regeneration. So when you damage your muscle, these are the cells that will actually repair your skeletal muscle. They're perfectly capable of regenerations in a healthy individual. But here we're talking about patients with cachexia, patients with sarcopenia. So the skeletal muscle function under these conditions, or under muscular dystrophy conditions, for example, a number of genetic diseases, declines. There are various reasons known for this decline in their function, but suffice to say that in the case of cachexia, for example, you have studies suggest that there's fiber breakdown and then that leads to muscle stem cell activation, but perhaps their differentiation into the muscle cells is impaired. We have some evidence, and again, this is the work that we do in the lab, that also suggests that there is impaired differentiation of muscle stem cells. So you have muscle stem cells, but somehow in the presence of cancers or the signals that are received from the cancer environment, these cells are not capable of undergoing differentiation to counteract the negative effect of cachexia. Because remember, these diseases result in atrophy. Atrophy is the shrinkage, reduction in the muscle. And so normally in a healthy person, you would expect that stem cells will kick in, they will activate, they will be mobilized to counteract this effect, but under this wasting conditions, that function becomes impaired.
So how does exercise come into the play? So when we, for example, lift weight, our muscles tear up, and usually after a good recovery day, they regenerate and we become stronger. Does those stem cell has a role in that kind of?
Absolutely. Yes. So exercise, there are two ways that you can grow your muscles with exercise. I'm not an exercise scientist, biologist, but one is through a hypertrophy, that is the increase in the muscle fiber without contribution from the stem cells. So you just get protein synthesis, you increase the fiber size, the myofiber size. That's through the hypertrophy. The other way that exercise can actually help you build your muscle is with exercise you break some of the old muscle fibers. So once they're damaged, then the stem cells associated with those or the neighboring fibers, they will actually get these activation signals as a result of injury, and they become activated and they contribute to muscle fiber repair. So you get both. You get muscle growth through hypertrophy, and then you also get fiber repair. Those are damaged muscle fibers in your body that are repaired. So
do we know if testosterone or steroids, hormone play a role in that?
Yeah. I'm not expert in this area, but yes, they do. Yeah. Absolutely.
A lot of people taking steroids.
Yeah.
So, how can we restore muscle stem cells' capacity to regenerate as we age, or in terms of cachexia?
Yeah, that's a great question, and if I had the answer, I would have.
What is your approach to that?
So the way we kind of when it comes to the aging, at least, what has shaped our recent, and by recent I mean within the past 10 years, our own research, our own direction, has been the realization and accumulating experimental evidence that you can actually restore, at least partially, the function of stem cells. And this doesn't apply just to muscle stem cells, but we're focused on muscle stem cells. That you can restore it. There are the ways to restore or to rejuvenate these cells. So some of the studies, they came out from many years ago, the parabiosis, where you connect the blood of the young and the old animals, and then it improves tissue regeneration. More recently, and what I mean recently is within the past 10 years also, that it's been shown that you can actually use the pluripotency factor to partially, reprogram cells, skeletal muscle stem cells, or any other cell type. And so the way it works is that there are 4 Yamanaka factors, right, that induce pluripotency. It was discovered that published in 2016 by Shinya Yamanaka. Now you can express these factors to bring a somatic cells back to the embryonic state, or you can actually do a partial reprogramming, and this is what is important in the context of epigenetic rejuvenation. And the idea behind it is that aging leads to accumulation of various epigenetic marks, and epigenetics is what controls your gene expression, right? How much of your gene A is going to be expressed will be dependent on the epigenetic marks that are deposited there. It's a complex process, so that includes DNA methylations, histone modifications, and a lot of other factors too. So you can use that by removing some of the age-accumulated epigenetic factors on your genes or your DNA, and then you can restore, you can let that cell to differentiate again into the cell type that originated from, right? Because it's partial, so it does contain some of the landmark that guides the cell towards the same lineage. It's not going to be randomly differentiated into any other cell type. So with those two in mind, partial reprogramming and then rejuvenation by connecting the blood of young and old, at least within the past 10 years, our approach to, I guess, rejuvenating or strategies to devise rejuvenating methods for the skeletal muscle has been, if we can find factors, these could be protein factors, could be molecules, could be anything in the skeletal muscle itself in the young that can have this rejuvenating effect on the aged stem cells. Keep in mind, organs, tissues, the way we look at it, these are communities, right, that contains billions and billions of cells. In this case of skeletal muscle, you've got myogenic cells, obviously, the muscle stem cells, the myofibers, but a lot of non-myogenic cells which are critical for the function of skeletal muscle. And then so these cells communicate, with each other. Without communication, a cell doesn't operate alone, singly. And so we think that aging, at least, it's been known that it dramatically changes this kind of communications. So then it becomes complicated, but then, trying to figure out how the cell-cell communication occurs in a young muscle or a young person, healthy person, versus what is caused by aging. And then trying to figure out if we can utilize that knowledge or leverage that knowledge to find out those factors which are important. And it's not going to be one factor, or it's going to be multiple factors. That will probably be important to try to use those to rejuvenate the skeletal muscle.
So by aging, like for example, is it because we become less active? Because for example, if you think about it in a mental perspective, sure, when people age, they become less mentally active and that kind of declines their mental activity. Is it the same with muscle dystrophy as well, or is it just because all the other factors?
Yeah, well, this question it looks straightforward, but it's a very complex question, right? So why do we age? There are all kinds of theories about it, but yes, with aging, all of our organs and tissues and cells, they actually decline in function. In the case of stem cells, there's also decline in their numbers, okay? So the question then becomes, are the cells that are lost in aging, are they any different than the cells that remain? We don't know the definitive answer to that. Why do we age? That's partially scientific, partially philosophical, partially something mix of everything else. So that's an interesting concept, but at least in the lab, we're not interested in that particular aspect. Yeah, but we do know.
So if you do some sort of things to not get old soon, for example, exercising or eating healthy, would possibly could stop those stem cells to be.
It'll slow. Absolutely. Exactly. Exercise and diet are very critical, not just for the skeletal muscle, but for the general health of the body too.
Okay. Yeah.
As of right now, do we have any medicine or drugs that can help with these two conditions or no?
No, for both conditions, exercise and nutritional regime.
Other than that?
No.
Okay.
No.
Interesting. So, it's a great field, and I'm sure Nika is going to enjoy it a lot when she starts her honors with you next year. So let me ask you now a little bit about teaching. So you also do teaching. Which one is your favorite? Or which one do you prefer?
Research or teaching?
I honestly, I'm very passionate about research. I do still go to the lab and then do pipetting and then do set up experiments. I still know how to do experiments. But I also like teaching quite a bit. Which one is my favorite? I think it's nice to do both. Sometimes, you just get away from thinking too much about molecules and then try to interact with students and
It's like good to mix it
yeah,
up.
yeah, absolutely.
What is your favorite part about teaching?
My favorite part about teaching,
Yep.
yes. Well, to be able to talk to people, because once, as a PI, when you're writing grants or writing papers or reviewing papers, you're in a crummy office, you don't interact with people. So at least that part, you interact with people. For me, because I'm a father too, teaching is almost like raising my own kids, not on that kind of a scale, but, it's both frustrating and rewarding, put it this way, but at the end, it gives you a lot of satisfaction.
Right. So would you say, well, let me put it this way. As an undergrad students, do you feel that they lack the knowledge when they come into your lab, or which type of student is your favorite? Sure, PhD students, they have more experience and stuff like that, but do you think there's a lack, or?
Yeah, so lack of knowledge is not a big issue. Lack of skill is not also an issue, because our job is to train people. Enthusiasm is what I look for in the students. So if they're enthusiastic, if they're curious, then they're the type of people that I would definitely, yes.
Oh, are you interested?
Of course. Otherwise, I wouldn't be here. Okay. I haven't personally taken your class, but according to what my peers have told me, you prefer to teach like more broader concepts and like critical thinking rather than just memorizing small details. How do you inspire students to take that role? To not just memorize, but think how they can like apply different concepts to a certain problem.
Yeah, so when it comes to that issue, people think that, if you have been teaching at the university for 10 years, 12 years, 15 years, then you have learned the routine, right? It's, at least the way I think about it, and a lot of my colleagues that I know, they think about it. Teaching is evolving field, just like research is. The way we teach now is different than the way we used to teach like 5 years ago, even 10 years ago, for example. When I was an undergrad, the source for scientific literature, you would go to the libraries like this, and then you would actually find these periodicals, papers, you would go through and find out. Then came the World Wide Web and the internet. For you guys, you're young and you think it's been there forever, but there was a time when these things were not there, right? Now we have the artificial intelligence, where you can actually ask this AI platform to summarize whatever you want to summarize, and then you get all that information. So in that context, then for me too, just like for students as well, perhaps more so for a professor, is constant struggle as to how do I change, how do I modify my approach to teaching to make sure that I'm not giving students redundant stuff, like things that you would be able to get from Google. This is why I don't emphasize on memorizing. There's no memorization in my class. I tell them in the beginning of the class that I'm going to give you a lot of information. Some of it I knew from before, and some of it I just looked it up yesterday as a refresher. But I don't expect you to really remember any of the details. So what I try to introduce in each class or in a course is our concepts that are evolving concepts, for example, fields that we know a little bit but not a lot about them. And then so I pose questions, and so you have this problem, we know this little or this much about it. How would you go about to figure out what is for example, if it's a disease, if it's a biological problem, how would you figure out what is causing this, or what is the mechanisms behind this? And there is no right or wrong answer, because the experiments have not been done. But what I try to figure out is if the students are able to think, and if I am able to make them think as a result of that. So yeah, there's very little memorization in my classes, and that's always been the case. It's mostly thinking. So.
Speaking of classes, which classes do you teach for undergrad, or do you even
Well, here I'm new, so the first year I was exempted from teaching, kind of. I had some guest lectures, and then this year I was teaching TMM 3107, I guess. That was 3107. Was also guest lecturing in other courses, BCH 8109, that's a graduate course. I still volunteer to teach 2 sessions, like 6 hours at McGill. That's the course I developed there, and then it's continuing. A colleague of mine now has taken over it, but I give them 2 lectures per year. I also coordinate the seminar course, the BMI master's and PhD courses with Professor Pellegrini. Yeah. So yeah, but at McGill, when I was there for about 9.5 years, I developed a course for our Department of Human Genetics. It's called Methods and Concepts in Chromatin and Epigenetics. That was a graduate course. So I was giving a lot of lectures, depending on the year, sometimes 5, 6, and then I had guest lectures. And then for a few years, I was also teaching another course for the Department of Medicine. This was for medical students, called Research Fundamental 1, 2. And this course was about, teaching or educating young med school students about, the drugs that you eventually are going to be prescribing to your patients. How does that end up here? Where did it come from? So basically going from the very concept of this basic biomedical research, in the lab and all the pipetting, down to the regulatory process of getting this clinical trials and whatnot, and then eventually developing into a drug. And that was fascinating for them, because they don't think about all that takes for a drug to be developed. So we had a lot of nice discussions.
Nice. Do you think McGill students are smarter or Ottawa?
Ottawa, of course, I'm in Ottawa. No, there's no such thing as. It really depends. I have seen very smart student here, and I've seen very smart student at McGill too. So there is no such thing. If you ask me about the food, I'll probably have a different opinion. But
Between the 2 institutes, which one was your favorite? If we factor your bias with Ottawa, you prefer Ottawa for sure, but between the 2 institutes, which one was your favorite?
So institutes, universities, cities, it really, when you say I like this institute or that institute, it's all the people there, right? Sure. You like it because of the people. I've been fortunate, to have great colleagues in both places. At McGill, when I went there, I was new, but very soon, I started collaborating with various colleagues there, and we still continue to collaborate. They were great people. Like I said, Ottawa was not a completely new place to me. I already know a lot of people here as a student, and as a postdoc. And when I came back, they have been just fantastic in terms of working with me, offering me if I need anything. So really, I've been fortunate to have these fantastic places people in both places.
So let me switch the gears a little bit now. What are your hobbies? What do you enjoy doing outside?
That's a tough one, right? Yeah. So when I was younger, I used to do a lot of hiking and mountain climbing and rock climbing too. I'm an outdoor enthusiast. I used to be camping almost every summer. Of going from Nova Scotia all the way to British Columbia, driving there. Yeah. And then US, that was when it was safe to go to US. Yeah. So now that I'm a dad, my son is 15 now, but for the last 10 years, he's into soccer, right? Competitive soccer. So I take him every week at least twice or 3 times to different places. He has practices, and then he has matches. So my hobby has become just basically going through soccer stuff with him. I do also. I read. I read on history. History is one of the things that I really like. Keeping up with current affairs, not much else outside of the, just taking care of my son and trying to fix things in the house.
Would you have a favorite book that you can suggest?
Well, I like, I think that the most recent one I had was a book written a while ago by Margaret Macmillan, Paris 1919, The 6 Months That Changed the World. I'm interested in history and how this politics plays into, and all past politics is giving rise to new conflicts, things like this,
If you weren't a researcher, what career path would you choose?
No contest. A farmer, yes.
Oh,
Yes.
okay.
Yeah, I love farming. I mean, I haven't been a farmer myself, but like I said, as a kid too, here too in Canada, we're blessed with all this vast nature. I love the idea of working outside in the field and producing my own food and raising animals, and it feels so cool.
So maybe for our last question, let me ask you, what is it that gets you going these days, and what is it that gives meaning to your life?
Oh wow, that's the toughest question, right? What is the meaning? What is the meaning of life? Okay, yeah.
Or what motivates you, or what gets you going?
Yeah.
What's the reason?
I look at life like a journey, right? It is a journey, and so when you plan a journey, when you go to, let's say you go to Europe, want to go to Italy, you have a list of things to do, right? Those are your goals. You want to go see this place. I want to go see this museum. I want to go see, you do these things. There's a list of things that all of them are your goals, right? And then so you do some of them, and then you somehow, something may happen and you're not able to do all of those activities. For me, I look at life in a similar way, like we have a very short span that we live on this planet. You have a set of goals. At the end of your life, you have achieved some of them, you have failed at some of them. What is left is a package that you have left there. So in that sense, life is really, at the end, it's not how much money you have accumulated or power or anything else, but how many lives you have touched, whether it's your kids, if they're happy with you, your wife, your husband, your students, the people there. So those are the achievements that I look at, how much have I achieved, which goals I have checked, done, and then which ones I have failed. And they're always both, right?
Cool. Well, I'm sure you're doing a great job, and
Thank you.
getting students like Nika, which I'm sure she's going to do great. And we look forward to having you again on our podcast, maybe on the campus, on the main studio, as you've probably seen.
Right, maybe on a Friday afternoon in a bar too, with a beer, not on a Monday morning, right? All right, yeah. Well, thank you very much.
Thanks again for coming to the podcast.
Yeah.
Thank you for listening to this conversation with Dr. Vahab Soleimani. We covered topics such as muscle wasting diseases, muscle stem cell role, partial muscle rejuvenation, and his teaching experience and life in general.
This is the Dendrite Podcast. To support it, please share this episode with your friends and anyone who might be interested. We hope you enjoyed this episode and see you next time.